name is Parinaz, and today I'm going to be telling you a little bit about my research on the origins of animal innate immunity. Today I hope to show you the motivations and evidence for pursuing this project, as well as the experiments that I'm currently conducting. As a tool to better understand animal evolution, I studied the closest living relative of animals, coenoflagellates. Coenoflagellates are globally distributed freshwater and marine eukaryotes. They've been found in a diverse range of environments, including the Arctic, Mono Lake, Strawberry Creek, the bottom of the seafloor, and so on. The coenoflagellate single cell on your right consists of an ovoid cell body, a collar of actin-filled microvilli, and a flagellum, which is used for propulsion when coenoflagellates are in the water column, or to generate a water column moving in this direction, outwards, in order to draw in bacteria to be phagocytosed. In addition to the unicellular form, some coenoflagellates are able to form simple, undifferentiated colonies, a likely precursor to multicellularity. All animals share a common ancestor that underwent a transition to evolve multicellularity. In order to better understand this transition, we study coenoflagellates, the closest living relative to animals. By comparing coenoflagellates to animals, we're able to identify genes that are shared exclusively between coenoflagellates and animals, and therefore reconstruct the gene content of the now extinct common ancestor of coenoflagellates and animals. This was likely unicellular or colonial. This comparative approach also allows us to reconstruct the early events in the lineage leading to animals, and better understand what genes were likely to have played a role in animal evolution. So what might this first animal have looked like? These comparisons between coenoflagellates and animals indicate that the common ancestor of animals was multicellular and had a simple epithelium. It likely possessed collar cells and preyed on bacteria, and probably was able to undergo apoptosis or programmed cell death, had an extracellular matrix, and may have been able to undergo some simple somatic cell differentiation. This is just a portion of the first animal, which was probably a hollow bone of cells. One aspect of this transition to multicellularity that particularly interests me is exploring what role the immune system may have played. Multicellular animals faced many challenges not shared by their unicellular ancestors. One such challenge was the evolution of mechanisms to defend against pathogens seeking to exploit these new niches present in the multicellular organism. As toll-like receptors play an integral role in the animal innate immune system, we think toll-like receptors might have been a good candidate to have evolved in the ancestral multicellular animal as a response to these pathogens. My project aims to lay the groundwork for asking whether toll-like receptors evolved in the earliest animals to answer this challenge. Toll-like receptors, or TLRs, are transmembrane proteins, or proteins that span the membrane of cells, and serve to recognize infectious agents. They're critical components of the innate or non-specific immune system, which is the first line of defense to pathogens and is thought to constitute an evolutionarily ancient defense strategy. Toll-like receptors are the best characterized group of pattern recognition receptors in animals. In mammals, toll-like receptors have essential roles in the direct recognition of infectious agents. Binding of microbial ligands to this extracellular leucine-rich repeat region activates the receptor and triggers a signal transduction cascade inside the cell that leads to the activation of transcription factor NF-kappa B and subsequent transcription of immune response genes. As I mentioned previously, TLRs have been well characterized in animals, which are shaded in green here. TLR-related molecules, meaning either the full receptors or the major components of TLRs, have been found in many animals, including the earliest branching animals, sponges. This has led to an understanding or a belief that TLR-related molecules originated in the common ancestor of all animals about 700 million years ago, and that toll-like receptors possibly originated 600 million years ago at the common ancestor of Eumetazoa, or all animals other than sponges. This is consistent with the belief that many animal innate immunity genes originated early in eukaryotic evolution, reflecting the ancient need for discrimination of self from non-self. But as this assessment compares only among living animals, it doesn't yield complete information on how the ancestral animal evolved or what was in its genome because of gene loss within lineages. For example, if a gene is found only in sponges, we can't tell if whether it arose only in sponges or whether it was in the common ancestor and was subsequently lost along the branch leading to Humanozoa. It's still unclear if toll-like receptors are present in sponges or could have been lost in sponges. So it could be useful to use an outgroup species, in this case, coinoflagellates. By sequencing a diverse group of coinoflagellates, we've been able to find what we believe are full toll-like receptors, which raises the possibility that TLRs may have originated earlier in evolutionary history and were present in the common ancestor of coinoflagellates and animals. What remains unknown is whether TLRs had an immune function in the common ancestor or whether they served some other role. As very little is understood about coinoflagellate innate immunity, characterizing the role of their putative TLRs, the goal of my research, could increase the breadth of knowledge about coinoflagellate immunity, especially in hypothesizing about the changing role of immunity in the context of the evolution of multicellularity. So here we have a phylogenetic tree of coinoflagellates that shows the evolutionary relationship between the 21 species of coinoflagellates that we have in culture. As I mentioned previously, in order to better understand where TLRs might have first evolved, it's useful to consult the next outgroup, coinoflagellates. Through sequencing these 21 species, we've discovered that 7 of them have what we believe to be full toll-like receptors. Of these 7, 3 species also had the downstream transcription factor, NF-kappa B. S. notiformis, Acanthoica spectacles, and Stephanoica diplocustata. Due to the presence of these 2 major components, we believe that these 3 species could be good candidates for further study of coinoflagellate immunity. So here we have a comparative view of the toll-like receptor pathway in animals and coinoflagellates. When looking at both the animal and coinoflagellate TLRs, you can see that they appear to be very similar and well conserved. While the actual receptors themselves look very similar, it appears that coinoflagellates lack many of the intracellular adapters and kinases that serve as intermediaries between the receptor and its downstream target, NF-kappa B. But this NF-kappa B is also conserved between coinoflagellates and animals. It's unknown if coinoflagellates possess different adapters or none at all. One possibility is that coinoflagellates possess a more ancestral and direct signaling pathway, whereby the receptor itself can initiate activation without intracellular adapters. As you can see in the second type of TLR that we have found in coinoflagellates, the intracellular region of the receptor itself contains a kinase, which looks a lot like this one. This kinase we typically find later in the pathway in animals. Therefore, one could hypothesize that animals having this coinoflagellates having this early form of a TLR didn't need the additional intracellular components and could signal more directly to activate NF-kappa B and begin transcription of the desired immune genes. You could imagine that as multicellularity evolved and cells differentiated, possibly opening up animals to a wider range of threats, it could be useful to have the ability to use a single transmembrane receptor whose intracellular region could interact with a number of different adapters and initiate a diversity of signaling pathways, leading to the development of these possibly newer intracellular adapters and kinases. So you can imagine that the coinoflagellate toll-like receptor is maybe a more ancestral form that may or may not have had an immune function and was later co-opted for use in the immune system to combat a number of different threats. So, in summary, these are the 2 major types of TLRs that we found in coinoflagellates. One resembling the canonical animal toll-like receptor organization, and a second novel organization that includes a protein kinase in the actual cytosolic region of the receptor itself, as opposed to a second protein within the cell as seen in animals. So how can we find out if these coinoflagellates also have a role in immunity as opposed to some other ancestral function? If coinoflagellate TLRs do indeed have an immune function, one possibility is that coinoflagellate TLRs signal in a similar manner to animal TLRs and have the ability to activate the downstream transcription factor NF-kappa B through a signaling cascade. One way to test this is to use the NF-kappa B luciferase assay that the Barton lab on campus uses. So how does this assay work? They first take human cells and transfect or introduce into the cell 2 plasmids, one containing a full-length clone of the TLR of interest that you'd like to test, in our case, each of the 2 coinoflagellate TLRs, and then a second plasmid that contains NF-kappa B and luciferase, a reporter gene. Luciferase is an enzyme that, when expressed, has the ability to bioluminesce or glow. So when NF-kappa B is activated in the plasmid, it will promote the transcription and expression of this luciferase gene, which will glow and acts as a recorder for when NF-kappa B is being activated, hence the name recorder gene. Once we have this TLR and recorder gene inside of the human cell, we can stimulate the cell with possible ligands, which are the components that the extracellular region of the TLR will recognize. That's right here. Once we've stimulated the system and hopefully activated the TLR and NF-kappa B, we will lyse the cells and measure the luciferase activity. If the TLR is actually being activated here and activating the NF-kappa B as well, luciferase should be expressed, and we should be able to measure the luciferase activity as a proxy for TLR activation. However, there exists a potential complication. As we discussed earlier, choanoflagellates appear to lack many of the intracellular components that make up the animal signaling pathway. Without these intracellular components, and possibly without the ability to bind mammalian intracellular adapters, these choanoflagellate TLRs might not be able to activate a mammalian system. One possible way to get around this problem and still utilize the assay would be to create a chimera, or a fusion construct of choanoflagellate and mammalian TLRs. You could fuse the extracellular region of the choanoflagellate TLR with the intracellular region of the mammalian toll-like receptor, which is the area that would actually bind to the mammalian intracellular adapters. In this way, you could still test for activation of the mammalian system and gain some insight into what types of ligands the extracellular region of the choanoflagellate TLR is binding, and possibly a clue about whether this TLR has an immune function or not. So future goals that I'm working on right now is to test full-length choanoflagellate TLRs and chimeric TLRs for activation of NF-kappa B in the luciferase assay. We could also find out what ligands the choanoflagellate LLRs are capable of binding, which should give us some clue about whether this is being used for immune function or some other role. And I also hope to discover the localization of toll-like receptors in choanoflagellates through staining with antibodies. Thank you, and big thanks to Dan, Olivia, and Ethan.